that cannot be seen by the naked eye. Now we go to the types. We start by the terminologies used in microscope. Terminologies. The terminologies. First we have magnification. Magnification. One of the terminologies. Magnification. So what is magnification? It is a process of enlarging the appearance of an object. Process of enlarging the appearance of an object. Magnification is the process of enlarging the appearance of an object. Now, the second one we have, this is the first terminology. The second one we have resolution. Resolution. What is resolution? Ability of the microscope to distinguish between 2 points that are close together. So it is the ability of the microscope to distinguish between 2 points that are close together. Ability of a microscope. This is resolution, the ability of a microscope to distinguish between between 2 points. Ability of a microscope to distinguish between 2 points that are close together. That are close together. That are close together. Now we allows for a detailed image. A higher resolution allows for a detailed image, while a lower resolution does not give a detailed image. Now this is about resolution. And now we go to the third one, that is specimen. Specimen. That is our third terminology. What is a specimen? A sample being observed and placed on a microscope slide. So a specimen is just a sample being observed and it is placed on a microscope slide. Let me write here. A specimen, it is a sample that is placed on a microscope slide that is used, that is observed. So number 3, we have the specimen. A sample placed on a microscope slide. On a microscope slide that is observed. So what is observed in the microscope? It is the specimen. We call it the specimen. The sample, we call it the specimen. Now we go to the fourth one. We have the slide. What is a slide? Terminologies used in the microscope. We have, it is a flat rectangular piece of glass where specimen is placed. This is where the specimen is placed for observation. Is placed for observation. So the slide, it is a place where the specimen, the sample, it is placed for observation. Now we go to the fifth, the cover slip. Cover slip. What is a cover slip? A cover slip, it is a thin piece of glass that is placed over the specimen on the slide to protect it and give it clear view. It is a thin piece of glass placed on the specimen on the slide to give it a clear view. Placed on the specimen on a slide. It is placed on the specimen on a slide to protect it, and to give it clear view. To give it a clear view. So we've said a specimen, it is a sample placed on a microscope slide that is observed. And then this slide itself, it is where the specimen is placed for observation. And then the cover slip is it is placed on the specimen on a slide to protect it and to give it a clearer view. Now we go to the sixth one, field of view. So the field of view, it is a visible area of the specimen when looking through microscope. It is the visible area of the specimen. Visible area of the specimen. So field of view is the visible area of the specimen when looking through the microscope. So visible area of the specimen. Now we go to the next one, the seventh. We have the total magnification. Total magnification. What is total magnification? So this is the overall magnification achieved by multiplying the magnification of the eyepiece by the magnification of the objective lens. So this is the multiplication of the total magnification. It is the multiplication of the magnification of the eyepiece, magnification of the eyepiece by magnification of the objective lens, So total magnification, multiplication of the magnification of the eyepiece by magnification of the objective lens. And then cover slip, it is placed on the specimen on a slide to protect it. This is one of the function, to protect it, and the other one to give it a clearer view. And now we have the slide where the specimen is placed for observation, and the specimen, it is a sample placed on a microscope for observation. Now we have to get a clear view and a drawing of the microscope. So the microscope, now let's know what it's that object. A microscope and its parts and the functions. So the microscope. In the microscope, we have parts like the eyepiece, we have the coarse adjustment knob, fine adjustment knob, we have the nose piece, we have the body tube, we have the mirror, condenser, we have the stage, we have revolving piece, we have the limb, then we have the stage, and we also have a mirror and a hinge screw. From the eyepiece, which is on top, and the base, which is on the bottom, which helps for the protection and for the for the stability of the microscope. Now we go to learn the functions of each of the following parts. Let's start with the eyepiece. The eyepiece. The eyepiece. What is the function of the eyepiece? An eyepiece has lens that contributes to the magnification of the piece objects under. It usually magnifies 10 times. So it has a lens that contributes, has a lens that contributes to the magnification. So eyepiece has a lens that contributes to the magnification. Usually magnifies 10 times. So an eyepiece usually magnifies 10 times. That is our eyepiece, which is on top of the, it is the first part of the microscope. Now we go to the second one, that is the coarse adjustment knob. Our second part is the coarse adjustment knob. Coarse adjustment knob. A coarse adjustment knob. It moves to the body tube up and down for long distances, bringing it into focus. So this moves, the coarse adjustment knob, it moves to the body tube up and down for long distances, bringing it into focus. So this just to bring the specimen into focus. The coarse adjustment knob brings the specimen into focus. The coarse adjustment knob, it brings the specimen into focus. Now we go to the second one, which is the fine adjustment knob. The third one actually, fine adjustment, which is below the coarse adjustment knob. Fine adjustment knob. The fine adjustment knob, it's the fine adjustment knob, it moves the tube and brings the image into fine focus. So this brings the specimen into focus, but the fine adjustment knob, it brings the image into fine focus. Brings the image, it brings the image into fine focus. So the fine adjustment knob is found below the coarse adjustment knob. And then we go to our fourth one, which is the body tube. The body tube. One of the parts of the microscope, we have the body tube. So the body tube holds the eyepiece, it holds the eyepiece and revolving nose piece. It directs light from objective lens to the eyepiece. So this one, it directs light. Body tube holds the eyepiece, holds the eyepiece. So the body tube, it holds the eyepiece and revolving nose piece. And revolving nose piece. So the work of the body tube, it's holding the eyepiece and the revolving nose piece. And then we have the revolving nose piece. Our fifth one, revolving nose piece. Revolving nose piece. So the revolving nose piece, It holds and brings objective lenses into position. So the work of the revolving nose piece is to bring the image, the objective lenses into position. Brings objective lenses into position. Revolving nose piece brings objective lenses into position. Now, what is the objective lens? What is an objective lens? Objective lens. What is the objective lens? It contributes to the magnification of the object. So its work is contributes to the magnification of the object. So the objective lens contributes to the magnification of the object, and then the revolving nose piece brings objective lens into position. And now we have the body tube, which holds the eyepiece and the revolving nose piece. The fine adjustment knob brings the image into fine focus, whereas the coarse adjustment knob brings the specimen into focus. Now we go to our next one, which is the arm. Our next part of the microscope. The seventh part, the arm or the limb. Our seventh part, parts of the microscope. Parts of the microscope. So the seventh one, we have arm or limb. We have the arm or the limb. It is for handling the microscope. The arm or the limb, which is for handling the microscope. Used for handling the microscope. Used for handling the microscope. The arm or the limb, it is used for handling the microscope. Now we have the stage. Number 8, we have stage. So what is a stage and what is its function? It is a flat platform onto which the slide with object is placed. This is where slide with object is placed. So in the stage, this is where the slide with object is placed. Now we have the 9th one, clips. The clips as one of the parts of the microscope. The clips hold the slide firmly onto the stage. Hold the slide firmly onto the stage. Hold the slide firmly onto the stage. Now we have the 10th one, that is the condenser. Is one of the parts of the microscope. Condenser. So what is a condenser and its function? Now, condenser concentrates light onto the object. Concentrates light. The work of the condenser, it's concentrating light onto the object. Onto the object. So, condenser concentrates light onto the object. And now, the 11th one, we have diaphragm. We have a diaphragm. Diaphragm. So what is the work of a diaphragm? Diaphragm regulates the amount of light passing. It regulates the amount of light passing. The amount of light regulates the amount of light passing. So the work of diaphragm, it regulates the amount of light passing through the object. Now we go to our 12th. After diaphragm, we have the mirror. What is the work of the mirror? The mirror is one of the parts of the microscope. So the mirror, it reflects light into condenser. Reflects light onto the condenser. Reflects light onto the condenser, which the condenser concentrates light onto the object. Now we have a base. We have a base is one of the parts of the microscope. The base is used to provide support to the microscope. Provide support to the microscope. So the base, it provides support to the microscope, the lower part of the microscope. And then we have a hinge screw. So our 14th one, it's a hinge screw. What is the work of the hinge screw? This one fixes the arm to the base. Fixes arm to the base. Fixes the arm to the base. The hinge screw, it fixes the arm to the base and allows for tilting of the arm. Another one allows tilting of the arm. It allows tilting of the arm. Now these are the parts and the functions of the microscope. Parts and functions of the microscope. Now we go to the care of a microscope. Our next one is the care of the microscope. From the parts of the microscope, after knowing the parts of the microscope, how to take care of the microscope. So our next one is the care of the microscope. Our next subtopic, it's the care of the microscope. The care of the microscope. The care of the microscope. So number one, in the care of the microscope, always keep it away from the edge of the bench when using it. Keep away from the edge of the bench while using it. You should keep the microscope away from the edge of the bench while using it. Our second one, always hold it with both hands. Always hold it with both hands. You should handle a microscope with care. Hold it with both hands while carrying it, moving it around the lab. Now, our third one is clean the lenses with special lens cleaning paper. Clean the lenses with special lens cleaning paper. While cleaning the lenses, you use the special lens cleaning paper. And now we go to our fourth one. Make sure that the low power objective lens is in position in line with the eyepiece lens before and after use. So the low objective should be in position. The low objective should be in position or in line with the eye, before or after use, Before or after use, you make sure it is in line with the eyepiece lens, before or after use. The fourth one, you store the microscope in a dust-proof place free of moisture. Store in a dust-free place, in a dust-free place and free of moisture. So this is how you care for the microscope. Now let's go to the cells seen under the microscope. But before that, we talk about the types of the microscope. We go to the types of the microscope and the cells seen under the types of the microscope. The types of the microscope and the cells seen under the type of the microscope. The type of the microscopes and the types of the microscope. Type of the microscope. The type of the microscope. We have two types of a microscope. The first one we have light microscope, and the other one we have electron microscope. These are the two types of the microscope. The first one, light microscope, and electron microscope. Now let's talk about the light microscope. The light microscope. The light microscope, it uses light. It uses light. From the word light microscope, we find that it uses light, the visible light. This is the visible light. In order to see through the light microscope, we have to have the visible light. And now in electron, it uses the electron beams. Here it uses the electron beams. So in light microscope it uses light, while in electron microscope it uses the electron beams in order to see the objects under the microscope. Now let's talk about the differences between the light microscope and the electron microscope. The differences between light microscope and electron microscope. So in the types we have the differences between Now here we have the differences between light microscope and the electron microscope. So what is the difference between a light microscope and an electron microscope? The first one is the light microscope uses light. It uses visible light. It uses visible light. The light microscope uses visible light and lenses to magnify the image. And lenses to magnify the image. To magnify the image. If the light microscope uses visible light and lenses to magnify the image, the electron microscope uses electron beam and electromagnetic lenses. Electron beam and electromagnetic lenses. So, light microscope uses visible light, while electron microscope uses electron beam and electromagnetic lenses. Now we go to the second difference. We go to the size of the microscope. Now, light microscope is small and easy to use. Small and easy to use, while in electron microscope, so it will be the opposite of the light microscope. So electron microscope, it is very large and need special rooms. Very large and needs special rooms. So the electron microscope is very large and needs special rooms, while the light microscope is small and easy to use. Now, we go to the cost. The light microscope is affordable and available in most schools. Affordable and available in most schools. While, so the electron microscope, it will be the opposite of affordable and available. So it is very expensive and it is used in special labs. Very expensive. It is very expensive and it is used in special labs. So the electron microscope is very expensive and it is used in special labs. And now we have the uses. So light microscope, it is used in studying cells, tissue, and small organisms. Used to study cells, tissues, and small organisms. So, light microscope, it is used to study cells, tissues, and small organisms, while the electron microscope, it is used to study very tiny parts of cells. Study the very tiny part, of the cell. So this is used to study the cells, tissues, and small organisms, while the electron microscope, it is used to study the very tiny part of the cell. And now, in light microscope, it sees living organisms. Used to see living organisms. But in the electron microscope, it is only used in dead species. Used in dead specimen. So in electron microscope, the specimen are dead, while in light microscope, they are living organisms. Now let's go to our next part, which is the cells seen through the light microscope. Cells seen through the light microscope. This light microscope. Cells seen through the light microscope. we are going to discuss the functions of the cells that we are going to find in the light microscope. So our topic will be cells seen through light microscope. So we have the cells seen through light microscope. We have cells. We have a plant cell, and we have an animal cell. The cells seen through the light microscope, we have the first one, an animal cell, and the second one is a plant cell. So let me draw here I'll start with the animal cell, which is so the animal cell, this is a just simple animal cell that is, so we have the cell membrane. In the animal cell, we have the cell membrane, and then we have cytoplasm, and then we have the nucleus, which is here, nucleus. This is cells seen through the light microscope. And we have the other one, the plant cell. The plant cell, we have the, let me draw here, the plant cell, and the plant cell seen through light microscope. So we have, this is the cell wall. This one is the cell membrane. We have the nucleus. We also have tonoplast. We have the vacuole. We have the chloroplast. I think I've written somewhere. Chloroplast. The chloroplast. We also have the cytoplasm. Cytoplasm. So these are the cells that are seen through the light microscope. We have cells seen through the light microscope, and the others are seen through the electron microscope. This is a plant cell seen under a light microscope, and the other one is an animal cell seen through the light microscope. Now let's talk about the functions and its structures, the structures of the cells seen under the light microscope. Now we are going to start with the cell membrane. The cell membrane. So what is a cell membrane? Our first one, we have the cell membrane. So what is a cell membrane? It is a thin membrane enclosing cell contents. A thin membrane enclosing cell contents. So a cell membrane, it is a thin membrane enclosing cell contents, and then its function, it controls the movement of substances in and out of the cell. So it controls movement of substances in and out of the cell. So a cell membrane, it controls movement of substances in and out of the cell. Now, the second one is a cytoplasm. Cytoplasm. So, a cytoplasm, it is a jelly-like substance where chemical processes are carried out. So, chemical processes are carried out here, are carried out in the cytoplasm, a jelly-like substance where chemical processes are carried out. It is scattered all over the the scattered structures are called organelles. So the organelles, they are scattered structures found in the cytoplasm. The cytoplasm. And now we go to our third one, that is the vacuole. A vacuole. So the work of the vacuole, it is for storage. That is, it stores water, nutrients, and waste products. It stores water, nutrients, and waste products. So a vacuole, it's used for storage of waste, water, and nutrients. And then we have the cell wall. A cell wall, it is the outermost boundary of a plant cell. Outermost boundary of a cell. So cell wall, it is the outermost boundary of a cell. And then we have the chloroplast. The chloroplast that is found in the plant cell. Chloroplast. So the chloroplast, with special staining technique, it is possible to see the chloroplast. This with staining techniques. Chloroplast can be seen. So the chloroplast, it is found in the plant cell, and it is possible with special staining techniques, it is possible to observe the chloroplast. Now we have the fifth one, that is the nucleus. The nucleus, it acts as the control center of the cell. So, control center of the cell. So, the nucleus is the control center of the cell. It contains the genetic material. So, in the nucleus, we have the RNA and the DNA. So, it contains the genetic material. In the nucleus, we have the genetic material, which is the DNA and the RNA. And then we go to our seventh one, that is the tonoplast. Our seventh cell seen under the light microscope. We have the tonoplast. So a tonoplast. So the tonoplast, it's the membrane that surrounds the vacuole. So membrane that surrounds the vacuole. So the tonoplast, this is the membrane that surrounds the vacuole, and then it controls the molecules, the movement of the molecules in and out of the vacuole. Controls movement in and out of the vacuole. It controls the movement in and out of the vacuole. So the others are the cells seen under the electron microscope. We have the cells seen under the electron microscope. So after the cells seen through the light microscope, we have cells seen under the electron microscope. So here we just change from light to electron microscope, cells seen through electron microscope. The first one, we have the Golgi bodies. The Golgi bodies. So cells seen through the electron microscope, we have the Golgi bodies. They are thin plate-like sacs arranged in stacks distributed in the cytoplasm. So they are found in cytoplasm. The Golgi bodies found in cytoplasm, and then its function is packaging and transporting. Packaging and transporting packaging and transporting glycoproteins. And also the Golgi bodies produces lysosome. So this has been the cells through the electron microscope. We have talked about the cells the cells through the light microscope. We have talked about the care of the microscope, the parts of the microscope. We have seen the object, the microscope itself, the drawing. We have talked about the cell, the introduction to it. We have talked about the differences between the light microscope and the electron microscope. We have talked about the terminologies in the microscope, the parts of the microscope and its functions, then the care of the microscope. Now, I am going to give an assignment on the cells seen under the electron microscope, which we are going to discuss in our next lesson. So the next lesson, I hope it shall be a walkover. The cells seen under electron microscope and its function, the structure and its function. This has been Elimu Live with Mwalimu Gladys Musenya. I hope and trust that you have written down something and you have understood the introduction of the cell, the microscope, the parts of the microscope, the terminologies, the drawing of the microscope, the differences between light microscope and electron microscope, the cells seen through light microscope, the animal cell and the plant cell, the difference between the animal cell and the plant cell. And also, thank you for watching KUTV, Elimu Live, Mwalimu Gladys Musenya.